In our last lecture, we talked about the immune system, and I introduced it in terms of what the job of the immune system is, and I talked about the fact that there's really two divisions to the immune system in the human body. There's the innate immune system, and then there's the acquired immune system. Like I said in the last lecture, the innate immune system is what you're born with, and it's generalized. It's not specific to any type of pathogen. It just really is defending against anything that is going to be foreign to the body. Now, the acquired immune system, on the other hand, is what we're going to be talking about in this lecture, and that is one that you develop over your lifetime as you're exposed to different pathogens. So let's get started. So, what is the acquired immune system? Essentially, this is our immune system, as I just said, that's developed over our lifetime, and essentially what it does is it determines self from non-self. So what do I mean by that? Well, our body is made up of all these cells, as I've talked about many times. We're a community of cells. And the way cells are able to be identified from each other is they have markers on the outside of them. In the cell membrane, there's these embedded proteins, and on those proteins are these carbohydrates, which are going to basically be the identifying factor for your cell. And it lets your body know, hey, this belongs here, or this doesn't belong here. Now, we have a lot of different types of cells inside of us. Some of them are ours, and some of them are actually foreign. So, for example, our digestive tract is covered inside and out with bacteria. And that bacteria is good bacteria. They call it the microbiome, right? That's going to be what is responsible for a lot of our digestion. It actually affects our health. It affects our mood. And we want that bacteria there. We don't want our body fighting it off. Our immune system will determine whether something is supposed to be there or something is not supposed to be there. And it does that with a couple different ways. So how do we know if something belongs or doesn't belong? Well, that comes along with something called an antigen. So what an antigen is it's some molecule that's going to cause an immune response. It's going to elicit some type of an immune response. And this could be normally a protein with a carbohydrate attached to it, a glycoprotein or a glycolipid. And all a glycoprotein or a glycolipid is a molecule that has a sugar component and a protein component for a glycoprotein, and a glycolipid would be the same thing but with a fat and a sugar. And that's just so it could be carried in our blood. Our blood is water soluble, so glycoproteins and glycolipids allow the blood to transport things that are not water soluble, like, for example, lipids. So if you take a look at this image here, this shows you a pathogen, right? And then on the pathogen are these antigens. So antigens are normally going to be, like I said, a carbohydrate, and that is how our cells identify whether or not they belong. Now, the coronavirus, for example, you keep hearing about this spike protein, right? That's what you're hearing all over the place. There's this spike protein, and then we're building a vaccine which will attach to the spike protein and essentially get rid of the virus. Well, the spike protein is the antigen, and that's all an antigen is. It's some marker on the cell which allows our body to be able to identify it. What makes up the acquired immune system are going to be lymphocytes. So these are going to be lymphocytes that we talked about in the unit on blood that we just finished up. And where do these come from? They come from our bone marrow. It's a process called hematopoiesis, and basically our bone marrow develops all of our different blood cells. But the two that we're going to focus on here are the B and the T lymphocytes. Those are really what compose your acquired immune system. The neutrophils, the basophils, the eosinophils that we talked about in the blood unit, those aren't the acquired. Those are going to be your innate immune system. This is the specific immune system. So let's go through the differences between them. I'm going to start by talking about the T lymphocytes. These are cells that get developed during prenatal development, and they form in the bone marrow, in the red marrow of the bones, and they get released into circulation. Makes these cells a little bit different than your other blood cells and part of your immune system is they don't stay in the circulation. They actually migrate to the thymus gland. And your thymus gland sits right here. It sits like essentially right around where your heart is. And it has a couple different jobs, but one of the main jobs it has is this is where these T cells go to mature. So they are not in their mature form when they are first made by the marrow. They enter the circulation, they head to the thymus gland where they stay for a good amount of time, and there's a process that is undergone in the thymus gland that matures them into essentially their adult form. And once their adult form is established, or once they become mature, at that point they're going to go back into the blood supply, and they make up almost all of the lymphocytes in your blood. So we have another type, which we're going to talk about in a minute, called the B lymphocytes. But the T lymphocytes make up close to 70 to 80% of the lymphocytes in your blood. And they also are going to accumulate and stay in the lymphatic system. So in your spleen and in your lymph nodes, that's where these are going to end up. And they're going to kind of just hang out there until they're needed. So there's actually 3 types of T cells, and I'm going to briefly explain the differences between them and talk about how they work. So the first one you may have heard of before, it's called a helper T cell. So essentially T cells are activated when, like for example, a macrophage. So we talked about macrophage in the last lecture. These are those large phagocytic cells that just kind of travel through your tissues, and when they see something that doesn't belong, they will envelop it and they'll digest it. But they do something really cool. They actually will take a piece of, so let's say they ingest a virus. They take a piece of that virus and it ends up on the outside of the cell membrane of the macrophage. So it would be like if I ate a sandwich now and a piece of that sandwich, like after I digested and everything, a piece of that sandwich kind of just appeared on my arm or on the outside of my body. And then everybody else that walked by could see what kind of sandwich I ate. Now, obviously that's kind of ridiculous, but that's essentially what happens. So the macrophage will ingest this virus or bacteria, and then there'll be a piece of that virus or bacteria on the outside of the macrophage, and then the T cells are going to identify that from that. So they essentially get triggered by something being ingested, and that activates them, and they basically are going to do a couple different things. They release these chemicals called interleukins, and what they do is they cause B cells to jump into action. So basically the helper T cells are messengers. They let the B cells get rolling. And I'll talk more about that, obviously, farther on in the lecture. The second type are called cytotoxic T cells. And these are the T cells that are actually going out and getting rid of particles that don't belong. They secrete a compound called perforin. And what they do, the perforin is a lot like, there's a lot of similar chemicals. They're essentially all enzymes, and this cuts holes in the membranes of cells that have the antigen that doesn't belong. So for example, if it's this virus that we're talking about or any bacteria, these cytotoxic T cells are going to be what actually go out and kill it and attack it. So that's essentially their job. And then finally, there are memory T cells, and these are cool. What happens is they kind of just hang around after everything's over, and they continue to replicate, so they're always there. And then the next time you're infected by the same, with the same antigen, because now there's a marker for it. Now your body knows that that doesn't belong, and you're able to quickly attack it. So that's why vaccines exist, right? That's what vaccines are all about. You're exposing your body to the antigen, and your body's able to form this immune response, and then after your body fights off this vaccine, which isn't the actual virus itself, it's just the outside coat with the antigen on it, and I'll talk more about this in class. I'll explain how the virus plays into all this. Your body holds on to that memory, and it's, to me, that's just so awesome that your immune system is that effective, that you can not only get rid of something, but then your body can remember it, so the next time you're infected by it, you can attack it and get rid of it right away. So this is just a diagram that shows a little bit what I just said here. So if you take a look, you can see a virus gets into the body. A macrophage is going to ingest it, and then once it ingests it, it has these little pieces of that virus, which is what the antigen is. And then what happens is the helper T cell has a little receptor on it, which is built to attach to that virus, antigen, and that triggers the release of this chemical called interleukin. And interleukin does 2 things. One thing it does is it triggers the B cells to get working, which I talked about a minute ago, and then it also triggers these cytotoxic T cells to go out and start just getting rid of the virus. And it also triggers the replication of more helper T cells. So this is, it's called, basically this process is going to trigger more of it. That's that positive feedback that we talked about earlier this year. It's going to trigger the continuation of this process. Okay, and then the second type are called B lymphocytes. And B lymphocytes are very similar to T lymphocytes, and the difference is where they develop. So these are also made in the red marrow, but the difference between these is they don't have to go to the thymus. That's where the letter T comes from. T stands for thymus derived. So your T cells go to the thymus gland to become mature. The B lymphocytes do not have to. They just go right into the blood supply. And there's not as many of them. They're only about 20 to 30% of the circulating lymphocytes. So most of the lymphocytes are T cells, and then about 20 to 30% are going to be B cells. And they also are going to end up migrating to the lymphatic system. So they're going to be in the spleen, and they're going to be in the lymph nodes. And they hang out with the T lymphocytes. B lymphocytes and T lymphocytes work together. They're really part of the same mechanism. There's two types of B lymphocytes. The first one is called a memory cell, and they work just like the memory T cells. They get activated by T cells, I just mentioned that, so the T cells are what give them the information, and then they stay in your system, so the next time you get infected by that same virus or that same bacteria or whatever the pathogen is, those memory T cells are already there, and they're ready to start churning out the antibodies to fight off infection. And then finally, we have our plasma cells, and these are, when you hear about antibodies, that's what these are. So basically the plasma cells, their job is they produce these things called antibodies. And the antibodies are specific to an antigen. So for example, COVID-19, let's say, for example, I got the vaccine. And what the vaccine is it causes your body to have just the antigen. So it's not the actual virus. That's why vaccines are so amazing. You're infecting somebody, excuse me, with essentially the uniform of the virus. It's the part of the virus that lets your body know it's there, but it doesn't have the actual DNA that the virus, or RNA, depending on the type of virus, that is going to infect the cell. And what happens is your body is going to then mount an immune response against it. And it does that by producing these things called antibodies. And an antibody is basically just a small molecule that attaches to the antigen on the virus or the bacteria. And antibodies really have three main effects. The first one is they will directly attack the antigen. So what happens is they clump together. So one antibody can cause a bunch of different antigens to clump together and basically just make it so they can't move. So think of them as glue. So if I have a whole bunch of viruses in an area, the antibodies are going to make them all clump together and not allow them to do what they want to do. The second thing is called activate complement. And it sounds like it's a weird phrase, but basically what it means is it activates these proteins on the cells, which allows the phagocytic cells, the neutrophils, the macrophage, to be able to do a better job. Basically, it just slows them down so that the phagocytic cells can go after them and get rid of them. And then finally, they also increase inflammation. And with increased inflammation, you get more cells to the area to fight it off, and you're going to have a better immune response with inflammation. Inflammation, like I said before, is what is the uncomfortable part of infection. That's why you feel pain when you're infected. If you have an infection, it hurts. It's from that inflammation. So this diagram just is, again, similar to the last one. You have your virus that has the antigens, right? So the virus has these antigens. And what happens is that activates the B cell. So the B cell is not activated until the antigen is there. And what that does is it triggers these things called lymphoblasts. And I know I didn't go into detail on that, but those are the cells that churn out B cells. So you get these B cells activating, and there's two types. The plasma cells, which produce the antibodies, and the antibodies are going to be what actually prevent that virus from being able to do what it wants to do. And then you have the memory B cells, which just basically, they hang out and they wait and they keep track of this. So if you get this virus again, you already have the antibody ready to go. So you're not going to get sick because you're going to be able to squash this virus before it's able to replicate enough to cause a real problem. And the way an antibody works is they attach to these antigens and it just literally slows them down. So that's a very brief explanation of the acquired immune system, but it's really an amazing process. So thank you very much.